We would like to begin by acknowledging that Carleton's campus is on the traditional, unceded territories of the Algonquin Nation. Ottawa is home to many Indigenous peoples and communities who represent other diverse, distinct, and autonomous Indigenous nations. The Carleton community recognizes the continued responsibilities it holds as individuals and as representatives of an educational institution towards engaging in active reconciliation. Good evening, everyone. My name is Maria DeRosa. I'm Interim Dean of Science here at Carleton University. On behalf of my colleagues in the faculty, I am delighted to welcome you to the 2022 Discovery Lecture. Established in 2002, the annual Discovery Lecture provides an opportunity to showcase excellence in science journalism and is hosted jointly by the Faculty of Science and the School of Journalism and Communication. Its purpose is to showcase and promote excellence in science journalism by focusing on communicating science in a fun and interesting way. For many years, we've had a very positive and fruitful relationship with the Carleton's Faculty of Public Affairs and in particular, the School of Journalism and Communication. In addition to our annual Discovery Lecture, we have partnered to offer a concentration in health sciences for undergraduate journalism students, a program unlike any other in Canada. In science, we understand the important role that clear and impactful communication plays in relaying the relevance of our research to society, and we continue to incorporate communications into our programs. For example, our Bachelor of Interdisciplinary Science and Practice and our new certificate programs in Science and Policy and Science Communication are unique and enhance our students' skill sets in communication and community engagement. We are excited to continue to collaborate with the School of Journalism and Communication to the benefit of our students, our researchers, and our society as a whole. The Discovery Lecture has hosted many notable speakers through the years, including Jay Ingram, Canadian author and broadcaster whom you might recognize from the Discovery Channel's popular television show Daily Planet, and Bob McDonald, one of Canada's best-known science journalists and the host of Quirks and Quarks on CBC Radio. Now, I would like to invite our award-winning science journalist and Carleton University CTV chair in digital science communication, or journalism, Professor Sarah Everts, to introduce our 2022 Discovery Lecture speaker to you this evening. Sarah, the floor is yours.
Hi, thank you, and welcome everyone. I'm absolutely delighted to introduce tonight's speaker, Antonio Regalado. He's the senior editor for biomedicine at MIT Technology Review magazine. But before spending the last 2 decades as a journalist, Antonio got a Bachelor of Science in Physics from Yale University, and then he went on to New York University to do a master's in journalism. Then he went to work for the Wall Street Journal for 9 years as a science reporter, and then moved to Brazil, to São Paulo, to cover science, technology, and politics across Latin America for a variety of publications, including Science magazine. Now he's based in Cambridge, Massachusetts, where he primarily reports on how technology is changing medicine and biomedical research. So this means he covers issues that triangulate science, ethics, and business, from anti-aging remedies and that burgeoning field to innovations in lab-grown organs. But tonight, he'll be focusing on gene editing, talking to us about COVID lab leak theories to the CRISPR baby controversy, which I hasten to add is a story that he broke in 2018, beating out all sorts of media, including the Associated Press. So, Antonio, welcome. The floor is yours.
Great. Thank you so much, Sarah, for the nice introduction. You have expressed interest in my work and what I have to say for a couple of years now, and that makes me feel really good to be here and to give the Discovery Lecture, especially to the journalism students in the audience. And it reminds me that in dealing with scientists, like I spend most of my day calling scientists or chasing down scientists, it's really, my appreciation for their work, my sincere interest in what they're doing, which allows those conversations to occur and my reporting to happen. So I just thought that was a nice thing to think about. I'm going to be toggling a little bit between slides and just my own Zoom image here. So I'm going to just show one slide here to just introduce today's talk, which is titled Dangerous Genes, Dangerous Genes. And we'll be talking primarily, or I'll be talking primarily about these two stories. And the first, as Sarah already mentioned, was this scoop, kind of a big scoop of my career in 2018. Exclusive Chinese scientists are creating CRISPR babies. We'll talk about, or I'll talk about how I chased down that story, basically. And then on the right, you can see an article that we've published recently. It's actually by a freelancer. So this is more of a topic than a particular story. And it is about this, what is called the COVID-19 lab leak theory. This is just the idea that maybe COVID-19 is actually the result of a lab accident, that it was caused by a scientific enterprise. And I have to tell you, this story is still going on, even today. Ferocious battles on social media involving journalists, scientists, all kinds of anonymous figures. It is really brutal and is turned into something quite difficult for journalists to deal with. And I'll talk a little bit about that as well. Oh, and then to finish, I'm going to bring these two topics together. I'm going to show you a link that is maybe science fiction, maybe one day science fact, that brings together the CRISPR babies and the threat of a pandemic. So in fact, these two technologies, these two different stories, we're going to see how they're going to be reconciled in some of the most far out thinking about where genetic engineering is going to lead in the future. So that's where we're going to end up. And I'm looking forward to that. If I can just stop the share, if I'm successful. Okay. So I want to preface the talk, Dangerous Genes, by saying that, for the most part, biotechnology is great, and we live in amazing biotech times. At Technology Review, we're often writing stories about a gene therapy that cures, a child with some devastating genetic disease. So that is mostly what biotechnology is about. It's about all these good things that it's going to bring in the future, cures and treatments and everything that there is to look forward to. Recently, Sarah mentioned there's also even the hope of reversing aging. Like scientists with their technology can take a cell, one cell, and they can reverse it to its youngest state. So now the hope is, could we do that to human bodies? So even longevity and who knows, immortality is possibly on the table. And of course, there's this stunning development of the COVID-19 vaccines, which are, easily the fastest medicine ever developed with an incredible global impact, billions of doses already distributed. And it's not just one vaccine, it is 6 or 7. So I just want to emphasize that, 99% of biotechnology is good and 99% of scientists are doing the right thing. But of course, journalists are drawn to the 1% that is bad, that is scary. And that's what we're going to be talking about today, that is dangerous genes. So in addition to the good things, we're going to be thinking about some of the possible bad and scary ones and what role journalists have in bringing those to light. So if I can just switch back to my share here. Yep. Let's see if I can. Okay. So I do, this is my conflict. This is what I call my conflict of interest slide. I work at Technology Review, and it's a magazine published by MIT. So in fact, I'm paid by MIT, and I work for in academia. And this is a general interest magazine, which is not really even about technology. I mean, it's not really about MIT per se. It's just about technological innovations wherever they may occur. You may have read the Harvard Business Review. That is about business. It's not specifically about Harvard. And the second part of my conflict of interest slide has to do with my own sensibility. And I don't know if any of you there used to get this magazine. You could pick it up in the newsstand. It was called Weekly World News. It was kind of a newspaper. It was printed on very cheap material. I think it is not published anymore, but I used to buy this with gusto because it was all fake news. And they really had, they could make up any story they wanted, and they really made up a lot of good ones, some of which involved science. So here you can see, Garden of Eden found, scientists regrow the tree of knowledge from a seed. So I put this here because this is sort of my sensibility as a journalist. I like these crazy, unbelievable stories. But of course, I work at Technology Review. So everything I write has to be very serious and very true. But I've become pretty good at threading the needle here and finding stories that could equally well appear in Weekly World News or Technology Review. They're hard to believe, they're incredible, but they're true. And so that's the sweet spot that I love to find as a journalist. And so here are just some of the strange, incredible, unbelievable, but true headlines that we have written about. Human-animal chimeras are gestating on research farms, babies, DNA IQ tests, a startup company that was going to save your mind by putting you to death, do-it-yourself gene therapy, and so on. So one of the underlying themes here is this question of playing God. I mean, you saw the tree in the Garden of Eden. It's kind of a cliche, scientists playing God. It's kind of a cliche, but it's also not. I mean, it is really the oldest story that we have about knowledge and what knowledge means and sort of the dangers of having knowledge. I mean, that is the story of Adam and Eve. It comes up again in Mary Shelley's Frankenstein. So that is the tension. And the reason it's relevant to biotechnology, let me just advance this. The reason it's relevant to biotechnology is that biotechnology is about the DNA molecule, is about the molecule of life. It's always astounding to think of, but you and I and all the ants and tigers and giraffes, I mean, every living thing has DNA as its program, okay? The DNA is the thread of life. It is the thread that joins all life. And biotechnology now offers us the chance to change it, to cut it, to edit it, to alter it. And so that's why it's so profound. That's why biotechnology is profound and why it raises this kind of playing God question. And here, you probably already read it. I've had it up on the screen, a quote by Edward O. Wilson, a very well-known biologist at Harvard who just died. And he was asked, are humans going to be able to solve the sort of existential crises of living together on planet Earth over the next 100 years? And he says, well, yes. The problem, is that we are products of evolution, right? We have Paleolithic emotions, medieval institutions, but godlike technology. We've been created by evolution and now have obtained the power to control it. And so that is really at the heart of these stories. So let me just stop share for a second. There we go. And of course, it is becoming easier to control DNA. And that is the big story on my beat. It usually goes by the name of CRISPR. CRISPR is a new way to edit DNA that was invented in 2012. I'm sure you've heard about it. There have been movies by now about CRISPR, tons of front page stories, tons of issues that have come up around CRISPR. And so I just wanted to mention briefly what CRISPR is. It was discovered in bacteria. It's a bacterial immune system is what CRISPR is. And bacteria has a way of spotting a virus and storing a little bit of that virus's genetic information in its own genome. And then it also has a special enzyme, the CRISPR enzyme, which can cut DNA. And so what the bacteria does is that as it spots it can spot an incoming virus, and it sends out this little memory cassette that it has to recognize that virus, and it's associated with this cutting molecule. So it finds the exact match, it matches a virus. If it finds a match, it cuts that virus, cuts that virus's genome. And that is what CRISPR does. It targets a gene very specifically, and it can cut it. And so in 2012, two scientific teams, one here, one in the US at the University of California, Berkeley, led by Jennifer Doudna, and another in Europe, led by Emmanuel Charpentier. They later won the Nobel Prize for this. They kind of reduced this system. They figured out how it worked. They reduced it to practice in a way. They showed how you could basically arbitrarily cut any strand of DNA that you want using CRISPR. And quickly after they did that, it was shown that it could work in all kinds of different cells, human cells, mouse cells, etc. So CRISPR was important. And just one journalistic detail, which is interesting. I don't know if everybody in the audience knows, but journalists, science journalists get like a tip off each week from the science journals, the big ones, like Science or Nature or PNAS. These are the big journals. And all us journalists, a week ahead of time, we get to know what's in them. We get a kind of a press release. And this is where sometimes we find out what we should be writing about. But it was quite interesting. I look back, and in 2012, when this amazing paper came out about CRISPR, this phenomenal technology that everybody has been using and everybody has been talking about, I look back at that press release from Science, and there was really no mention of this paper. So at that time, people just did not realize how important it was, right? Not even the editors of Science didn't even think to flag it to the journalists. So it took a number of months, 6 or 7 months, before kind of journalists start to cotton to the idea that there was something big happening and started writing stories about CRISPR. And I like this anecdote because it's kind of what gets me up in the morning, the idea that there might be huge journalism scoops, huge science scoops that are just sitting in plain sight. They're just sitting there in the scientific literature and no one has figured it out yet. And that's what I like to do. I wasn't the first to figure out CRISPR, but there's always that potential. So I find that really exciting. So what happened with CRISPR? Well, very soon people started using it in all kinds of laboratory experiments because it was so easy to use. And they started modifying animals. First mice, that was here at MIT. And then very quickly in China, they had a whole herd of genetically modified animals, sheep, goats, and then a monkey, okay? And as soon as the monkey happened, as soon as they edited this monkey, a lot of people who are close to the subject matter realized that, okay, humans are going to be next. And that was an interesting moment because before that, everybody had said that, genetically engineering humans would be wrong under any circumstances. The European Commission on Human Rights, the Journal of the American Medical Association had a position. Everybody said that it would be wrong. But they had said that when they couldn't do it, right? And so suddenly it becomes possible. Suddenly it becomes possible for a reason I'll tell you in a second. Suddenly it becomes possible. And so now, now we have to reconsider our view. It's not so easy to say that it's wrong when you can actually do it. And the reason it changes everything is that CRISPR is just more efficient. Basically, it's a more efficient form of genetic engineering. So you could potentially do your CRISPR change or your CRISPR edit to a single cell. And you couldn't do that before. You could do it to a single cell, and that single cell can include a fertilized egg, like the beginning of an organism. It's the first time you can actually effectively make genetic modifications to a single cell. So that opens the possibility suddenly of editing human beings. And as a journalist, I saw that monkey, the edited monkey, in my mind, I was like, I know this is going to be irresistible knowledge to somebody. Irresistible knowledge. They're going to reach for that, the apple on the tree of knowledge. They are not going to be able to resist this one. So I set about covering CRISPR and covering its inevitable advance towards the first gene-edited human beings. And I just wanted to mention briefly how we went about that. It was basically just reading the scientific literature. because people had started to edit embryos. There was a few papers, like someone tried CRISPR in a human embryo. And those papers themselves were extremely controversial because people are still not ready for this idea. And when we looked at these papers, we found that there was only about 11 of them. There was one from the United States, one from the UK, but there was nine or 10 from China, right? China. So we decided we really need to go to China to find what is happening. And so that's exactly what we did. Let me go back to my slides if I can. All right. Okay, so this is where the story ends up. This is Hu Jiankui. He's the Chinese biophysicist who actually created the CRISPR babies. So I ended up going to China to try and interview all these gene editors, all these people that were just editing the embryos, to find out sort of what their intentions were, what their plans were, and potentially to find the secret project, which I ended up finding because I met him. He did not tell me about the CRISPR babies, but he gave me enough information to start researching him and eventually find the documents that kind of disclosed his plan. It's something that was interesting was that Hu Jiankui, these are some pictures from his social media feed. Hu Jiankui had, it's a secret project, but not really, right? He actually told quite a number of people that what he was doing. He was trying to make gene edited children. And in particular, he was trying to cut a gene called CCR5. He was trying to cut this gene in the embryo in order to make children that were immune to HIV, because it was known by scientists that if you lack this gene called CCR5, people who lack this gene do not get infected with HIV at all, or at least not very easily. So he was trying to introduce this same change into the embryos to make children that were resistant to HIV. And he had reasons to do so. In China, there's a huge stigma against HIV, and they're not even allowed to use IVF. So he had a kind of renegade program to solve this HIV problem for them in a way. But I just show his slides here because he did tell a lot of people. He told these fellows, these Americans here that are in this picture. This is Michael Dean, one of his advisors. To the right is Steve Quake, very important guy at Stanford, who heads the Stanford, Mark Zuckerberg BioHub. Here he is with Craig Mello, a Nobel Prize winner at a meeting in China. Here he is with the head of a big US health provider called Gelsinger. Here he is with John Kerry. So, and here he is with Jennifer Doudna, the inventor of CRISPR. So he really got around, and he actually, a lot of people knew what he was up to. A lot of people knew what he was up to, but didn't say anything. So I thought that was interesting. The scientists didn't sort of blow the whistle. They were not, they were kind of on the fence about whether this was right or wrong. And Steve Quake, the scientist that I mentioned, his role in all this, he knew about it. His role in all this later came under question, and he gave a big interview to the New York Times. And the New York Times asked him, like, why didn't you say anything? And he said, well, who should I call? The science police? Who is the science police? I thought that was funny because I said, well, you could have called the journalists, right? You could have called me. I would have loved that. Oops. Let me just keep going here. Get back to my shared screen if I can. Okay. From current slide. Okay, so I went to China with this documentary crew that are shown here. And they're actually making a movie about this whole episode. I haven't seen the movie, and I don't know how I'm portrayed in it or anything, but I would want to give them credit for taking me to China. And I see that I'm going to be short on time if I don't speed up a little bit. So I just want to say, here's Hu Jiankui. Our story came out 2 days or 3 days before a big conference in Hong Kong, which was the Human Genome Editing Conference, the second Human Genome Editing Summit. And it was a huge meeting of scientists to decide, they were gathering basically to decide whether creating CRISPR babies would be right or wrong. But the whole thing, they got the huge surprise just before the meeting of finding out that He Jiankui had already done it. And the reception was, for a few moments it was positive, but then quickly turned negative. And here's He Jiankui walking across the stage. He actually showed up in the meeting. He explained himself. He gave a talk, and he explained that he had created twin girls with edited genomes. He Jiankui is now in prison, and the reason that he's in prison is he was convicted of practicing medicine without a license, basically pretending to be a doctor. But the real problem here was not so much the gene editing of the children, which scientists still believe might be useful someday to avoid certain diseases, but that he had done it prematurely. He had done it prematurely, and the way the edits had turned out in these children was kind of a mess. Some cells were edited differently than other cells. He didn't have the technology under control. It's too early to use this technology for this purpose. But he rushed ahead. We found out what he had done, and he was called to account, and now he's in prison. So that was a pretty dramatic consequence of this this example of dangerous knowledge. I want to turn to the Wuhan Institute of Virology and what is called the lab leak theory. This story also occurs, of course, in China. So China is a country with huge ambitions in all kinds of domains, including biotechnology. They are rushing forward in biotech, which makes people anxious. It's a very powerful technology, and here's a country that has sort of a different government than ours in the United States or in North America, and different values, especially when it comes to freedom of speech and other things. So this is a cause for anxiety. Just see where I am. So this is a very interesting situation, which is that the pandemic, the COVID-19 pandemic, arose in the city of Wuhan, China. And so it turns out that the city of Wuhan is also home to, I think, the premier laboratory in the world dealing with this kind of virus. It's a type of bat virus, a bat coronavirus. So they have a huge collection of these viruses or samples from bats right in the city of Wuhan. So that is the coincidence that is at the core of the question about whether the virus came from the lab. It is simply a matter of the coincidence that the laboratory is in the same city a few miles away from where the outbreak began. And this debate rages on today, and I have assembled here a kind of a sort of synopsis of what the situation is. And on the left, I have the zoonoti. This is short. This is people who believe in a zoonosis. Zoonosis is when an animal virus jumps to humans, a spread from animals to humans. And on the right, I have sort of the profile of the lab leakers. And so we're going to see why these two theories for where COVID-19 came from, a natural sort of spillover, maybe in an animal market selling wild animals, the kind of place where all kinds of different animals are together in close quarters, maybe unsanitary quarters, and a virus is just given the opportunity to try out new species that it might not normally have access to, including humans. So who is the typical zoonoti? Well, it's a virology professor. It's a senior virologist, as opposed to the lab leakers, which are, most typically sort of an anonymous, angry someone on the internet, which does not really reflect that well on the lab leak theory. Although there are some very prominent people who believe a lab accident is the cause of COVID-19, including the former head of the CDC, Robert Redfield. So in terms of the evidence, it's really, there isn't enough evidence yet on either side to say for sure where it comes from. And that's why the debate goes on. The Zunoti will say, well, the early cases, it turns out, were not all of them, but many of them were associated with one of these animal markets. So they say, well, that is a pretty clear evidence that maybe it's this market spillover scenario. On the other hand, the lab leakers can counter that and say, well, the lab is in Wuhan. So if you're looking for coronaviruses, I can tell you where there's a whole bunch of them. On the natural origin side, they can say that it's happened before. It's not the first time a virus has jumped from animals to humans, and it's not even the first time it's happened because of an animal market. The lab leakers on their end can say, well, lab accidents have also happened before. In fact, even this current virus, SARS-CoV-2, has already leaked, accidentally infected lab workers on at least one occasion, and probably more. So lab accidents definitely happen. This kind of virus can get out. And then we get to an interesting point. The Zunoti will say, well, there's similar viruses in the wild. The lab leakers can say, well, there's similar viruses in the lab. But the virology professor will say that evolution can create this kind of virus. Like, there's nothing, if you look at the genome sequence of the COVID-19 virus, they don't see anything in there really that evolution itself couldn't come up with. And evolution is great at coming up with all kinds of surprising things. Whereas the lab leakers, they're really seeing the human hand in this, right? Human handiwork. So it comes down to this issue of whether it is nature and just evolution can be responsible for this virus, or whether indeed it is human caused by people in some way. So part of the story here, even if we don't know yet where COVID-19's origin, what COVID-19's origin is, most people, scientists, will lean on the side of natural origins, but they can't really rule out the laboratory. But whatever the case is, what has happened in this story is it's shed an incredible light on the kinds of research that is going on in these high-tech virology labs. Like, this is one of the outcomes of this COVID-19 debate, is to shed light on this research in ways that is kind of uncomfortable for scientists and kind of uncomfortable for funding agencies like the NIH, which actually paid for some of the research in the Wuhan lab. And this research involves genetic engineering with these dangerous bat viruses. They have, it's not CRISPR, it's a different set of techniques, but they have techniques, even from the code of the virus, if they've sequenced it, even from the code, they can actually recreate that virus, right? They can look it up in a book, and then they have a technique to actually make it again in cells just from the data. In doing that, they can also easily change it, and that's what they've done. They have taken, you take a part from one virus and you combine it with a part from another virus. They have even taken the MERS virus, another dangerous virus, and added pieces from different bat viruses. And the reason to do this research, they say, is to understand what gives a virus the potential to cause a pandemic like COVID-19. The whole purpose is knowledge. It is knowledge of how viruses work, what allows them to adapt to a specific host, like us, what allows them to transmit easily through the air, like COVID-19. They want to find out these things. And to do it, they actually kind of create the potentially dangerous viruses. Like, to study them, you have to create them. And that is sometimes called gain of function research. And the spotlight that has been shined on that right now is probably one of the most important outcomes of this COVID-19 origins debate. Because this research, some people argue that it's necessary, but now we understand with this pandemic what its dangers are. If you create a pandemic virus in the lab, it could get out. And then what is the consequence? It could kill 5 million people. So the debate right now is actually whether all this kind of research is too dangerous. If this is a dangerous knowledge situation that we can't tolerate. And the sides are sort of concerned citizens, concerned scientists, and on the other hand, funding agencies, virologists with a lot of investment in this type of research and in learning more. So I have a quote here. If I can get back to my screen share. Okay. Oops, go back one. Okay. This is text from a Washington Post editorial written actually by a scientist at MIT named Kevin Esfeld. And he, in no uncertain terms, called out this research. This research that has been going on in Wuhan, funded by the United States. He says that to go out and try and identify these viruses out in bat caves and bring them back, or to engineer them, these days, because of the ease with which genetic engineering can occur, he says it gives thousands of people potentially the power to wield that as a weapon, or to learn how to enhance a weaker virus. So he's concerned that this knowledge, combined with sort of the ease of genetic engineering these days, is just a very dangerous situation. So in his lab, for instance, he said there's at least 5 people in his lab who would be able to take this information from a publication and actually make that virus. Now, they're PhD scientists, but it could be concerning to have this knowledge around because, someone gets up on the wrong side of the bed, someone's having a mental situation. It creates a power for individuals, potentially individuals, to create a pandemic virus, and who knows what they would do with it. It's unlike nuclear power, nuclear weapons, where you need a whole state enterprise, right? You need to separate uranium and so on. You need hundreds, thousands of people and a big budget to make the nuclear weapon. But unfortunately, for making a virus, you don't need all that. Potentially, one person can do it. And he ends this essay by saying he implores the scientists to stop. I want to end by just saying how these two ideas, the CRISPR babies and the pandemic viruses, come together. In fact, that day when He Jiankui was on the stage, I showed the picture of him on the stage, and he had to say what he had done, he was immediately followed by George Daly, who's the head of Harvard, he's the dean of Harvard Medical School. And George Daly had he's like, well, I don't like what He Jiankui did, but he said, I think as a society, we're going to move forward with human gene editing of genetically modified people. He said, we're moving forward with it. And one of the reasons he gave, and I thought this was very interesting at the time, and I didn't really appreciate it until COVID-19, he said, we need this technology as a kind of a fail-safe. We need to be able to change our own genomes as a fail-safe in the event of some terrible pandemic, of a black plague, of some kind of existential disease crisis that's spreading around like in a movie and killing everybody. One way to avoid that, or one way to counteract that, would be to make our children immune to that disease, just like He Jiankui tried to knock out CCR5 and make HIV-resistant people. George Daly was saying, we need this as society technology. We need this technology as a society to protect us potentially against future diseases. We need to be able to make people that are not subject to a given virus. And then inspired by those comments and that idea, I actually found, we wrote an article, there's a picture of a pig, because we wrote about a pig company that is actually doing this. They have a disease in pigs. It's like COVID-19 for pigs. It's called PRRS, I think. It's sort of like a COVID-19 thing that pigs have. Pigs live in sort of big farms. These are pigs being grown, to turn into pork chops and bacon. And so they're very susceptible to these respiratory diseases. And what this company is doing is it's making a huge herd of clone pigs that have all been engineered with CRISPR to resist this disease. They're knocking out a gene in the pigs that makes them resistant to this disease. They're actually knocking out the receptor that the virus uses to get in the cell. So right here already, at least in pigs, we have someone trying to implement this technology to engineer these animals so that they're resistant to this terrible pandemic of pigs. So looking forward, it turns out that this idea of making humans that are resistant to viruses is actually an important one among the sort of most far-sighted scientists. And this guy on the left, his name is George Church. He's from Harvard University. We write about him all the time because he's always doing something unusual and interesting. And of course, if we're talking about playing God, he fits the role perfectly, right? He looks like the one from the top of the Sistine Chapel. And he wrote a book about synthetic biology, which is sort of the field that CRISPR is part of, in which he said that the climax of all these technologies, the climax would be the production of human beings that are resistant to all viruses, including HIV. When he wrote this, there was no COVID-19, but it would include COVID-19. And this idea actually involves writing an entire genome. Scientists can write little genes, and they're working on writing big genes, and eventually, in the near future, they're going to be able to write an entire genome. They're going to be able to type it out on a computer and then print it and then turn it into a living thing, including a living person. So the idea here, if you could write a whole genome, and so you could have people who have like a genome that you made, you could make little changes here and there in the genome so that that person would not be ever infected by a virus. I don't want to get into the details of how it works, but that is a possibility. And I thought it was so interesting that George Church, this pioneering, farsighted kind of futurist of CRISPR, thinks that that is actually going to be the big outcome. Human beings no longer subject to diseases from any virus at all. So with that, I think I will end, and I would love to take your questions about anything that I've talked about, as well as any other subjects that you want to ask me about.
Great. Well, thank you so much, Antonio. And yeah, I'll just remind everybody who's in the audience that if you do have a question, please write it out in the Q&A box. I'll be reading the questions to Antonio and keeping an eye on what's coming in. As you put together your questions, I have a few for him myself. And I think I'd like to start off just by jumping off from what you talked about, which was like, after the news that the CRISPR baby had been born, or several had been born, there was, much ado, hair pulling, committees and panels struck to address the, ethical line that had been crossed, with people saying things like, there must be some way to police this science. I guess my question to you is, is there a way, and actually has anything been done? Or, is there just a lot of people pulling their hair? Any thoughts on that?
Well, I think, all the blowback that there was kind of reinforced the point that the technology is not ready, right? It's just not ready. It could be ready pretty soon. It's just not ready today. So I think that most scientists have sort of, they have stopped their march towards it and sort of accepted the notion that it's not time yet. There is a debate, one of the controversial things that actually George Daly, who I mentioned, said at the conference, he said, the debate is over. He said the society debate is over and we're moving forward. We're going to move forward in a scientific way, which means not now, but we're definitely moving forward. And some people didn't like that because they said, no, the society debate really hasn't occurred. But I think scientists realize that it could be medically useful. They have a little bit of a hard time finding the use cases. They're not that obvious. But, they fully intend, as a community, they fully intend to keep developing the technology and keep understanding it, and have reserved the right to themselves. They're reserving the right to use it. Although I should note, I don't know the situation in every country. In the United States, it's banned. You cannot. It would be illegal, impossible to make a CRISPR baby in the United States, which is its own problem because then maybe it moves forward in some place with fewer rules.
There's a question from an attendee just about the CRISPR babies. Does anybody know where they are, what they're doing now? How old are they? I mean, are they 4 or 5?
No. They were literally born like a week before this, maybe a month before our story, and a month and a couple of days before this conference, and when it all became public. So they are now a year and a bit. Is that 2019, 2020? Oh no, they're going on 4 or something. I'm not doing the math right, but yeah, they're toddlers somewhere, and there's really no information about them. I think there's 3 at this point, and there's 0 information. So that's one of the features of China, is that you can get a situation, they don't have the press. This is not a good subject, right? The press is not invited to find out more and to track down these babies. There's medical privacy issues, right, as well. But the fact that we know absolutely 0 and we'll never hear about it again is a little bit of feature of China's system.
Just to jump off of what you were saying before, there's a question from Candace Harris that says, with the earlier quote you mentioned, wouldn't urging people not to explore dangerous areas of science actually make it more likely? For example, doctors won't implant chips, and, there's lots of online forums with all sorts of weird biohacking ideas, I guess, or do you think that, that actually regulating does successfully control it? I mean, you kind of alluded to it, when you put regulations in place, things just go into the dark. Can you talk a little bit more about that?
Well, George Church, whose image was the last one we saw, he has actually said he thinks scientists should have to have a license to operate, like to do gene editing. And by license, he means, even someone who does, cuts dog's hair needs a license. Right. So who are you? We need to have your name, your number, your phone number. You need to have a license, maybe show that you've taken Ethics 101 or something. So at least let's know who these people are. But he's also said that he thinks that people changing genomes should be under surveillance. And he meant it. Surveillance by their peers, by their colleagues, by their universities, and maybe even by the government. I mean, it gets a little scary when you talk to some of these scientists at the frontier, because they know what power this has. And I just think it's not a debate that biologists in general want to have. It's not a discussion that they want to have, because, to do science is it's freedom of thought. It's freedom of expression. I want to find out this, or I want to do this. They don't have a tradition of being under heavy surveillance or control. On the other hand, a nuclear scientist would, right? I mean, if you have uranium, people are accounting for where the uranium is and how many centrifuges. It's all very controlled, and we accept that. But we don't yet accept that with biology, and hopefully we'll figure out some kind of approach before something bad happens.
So there's a bunch of questions just on the COVID-19 side of things, but before we jump over there, I just wanted to wrap things up with just gene editing writ large, because before CRISPR came on the scene, speaking as a science journalist, it almost felt like gene editing therapies or gene therapies was kind of a tired, old, hyped idea, right? It just felt like it had been, an idea for decades, and there had been not a lot of progress. Can you talk a little bit or tease out some of the challenges for why that was so hard, and any actual successes from more kind of traditional gene editing technologies?
Gene therapy, right.
Yeah,
gene therapy gets its start, I guess, in maybe the first attempt to do it is in 1991 or something. That involves, it's also a problem for CRISPR. You can do wonderful things in the lab, but the question is, how do you get it into a human body? And here we're not talking about a single cell that you can actually, plunk, you can inject it right in. We're talking about a child or an adult. And so the issue is what they call delivery. You have these genetic tools and you have these genetic instructions. How can you get them into the body? That is the big challenge. That has been the challenge for gene therapy, which I want to say, has come of age, and there's like now several gene therapy just in the last, since 2017 in the US at least, there's now 4, 5, 6, 7, 8 gene therapies in the market. So they're coming along, and CRISPR is just sort of, let's call it gene therapy 2.0. It's just an advanced type. So it's really this question of delivery, which has partly been solved, but there are a ton of companies working with CRISPR to try and cure inherited diseases of various kinds. And they're moving along quite quickly. I mean, the technology is still pretty new, but it's reaching clinical trials. There've been a few clinical trials, and there's going to be many more of them.
Also, the scientist who did the CRISPR babies, so there's a question in the chat. Have we heard much from him? Is he okay? Is he alive? Or is that all even a mirage and he's in a lab somewhere doing more experiments? Has he been heard of by anybody?
I mean, I only get things secondhand. As far as I understand, he was under house arrest for some time, but as I understand it, I haven't verified it, he's actually in prison. His prison term is not that long, it's something like 3 years. So is it 3 or 4 years? In fact, I need to go check. The questioner is reminding me, I got to go check if he's out, because he could have some kind of amazing second act. I mean, why not? He could have an interesting second act. I did hear that when COVID hit, there was a plan to spring him from prison to help, right? Like everybody, it was all hands against COVID, especially in China. And of course, he's an expert in genome sequencing and all the relevant technologies. So I did hear there was kind of a bid that he could get out to help, but that didn't happen.
Interesting.
Yeah.
So yeah, let's speaking of COVID, there's a question in the audience. Do you have a sense, like, do you have a gut reaction of, whether it was, what did you call it, zoonote or lab leak? Would you willing to go on the record as saying what you?
Zoonote, yeah, is the person that believes in zoonosis, like sort of like the Illuminati.
Yes.
Zoonote, yeah, right. I vacillate and I waffle on this, and that's why I'm blocked on Twitter by people on both sides of the debate. I haven't satisfied them by taking up a banner. I mean, I cover biotechnology, so in a way, the outcome that I'm interested in, or would be more interested in, is the laboratory accident, because then it would be due to technology, and it was also the one that I hope that I might be able to, or that someone might be able to actually discover, to prove the case through documents, right? Just like the CRISPR baby, I didn't get into details about the documents, but that was a document-driven story. It was equally possible with this lab leak that there could be a pre-existing document created by science or by scientists that would have solved the case, right? I mean, if you're driving one of your own viruses and you have an accident, you're going to leave parts of that virus around, so you might be able to make the connection. And so it was disappointing that so far, at least, the document hunt has not been, the most important evidence. It doesn't solve the case. I'm not answering the question because I want to reserve. I really don't know. I don't know.
So, many journalists who've covered the lab leak theory have been attacked online. I'm wondering, can you describe just writ large the current climate for journalists covering these topics? Are they being trolled? Share with us a little bit.
Yeah, it is absolutely terrible. It is absolutely terrible. We just published a Technology Review. We just published an article that was sort of from the point of view of this scientist in China, like the chief scientist of the Wuhan Institute of Virology. It was by a Chinese science journalist who speaks excellent English and is trained here, named Jane Q. And, ever since the article published, there has just been one gigantic flame war on Twitter with people questioning her motives, whether she's a member of the Communist Party, whether the government sort of, controlled what she said, why she didn't ask certain questions. And it's been, just extremely, extremely unpleasant. And it's unpleasant for me in a totally different way, which is that, scientists don't want to talk about this. It's so political. It's so nasty. Nobody wants to talk about it. Nobody wants to go on the record. And if they even have if you even, if they even suspect that you might be a lab leaker, then they won't even talk to you. So then they play, favorites with science journalists. Like certain people have access and certain people don't. So journalists have stopped talking to scientists. Scientists have stopped talking to journalists. Journalists have stopped talking to one another. People in China have stopped talking to people in the United States. So that's not a great situation.
Yeah.
Yep.
We have a question from Martin Holtzik about the evolution of pathogens. So, the evolution of pathogens is always going to be faster than that of the evolution of humans, right? They procreate faster, or divide actually faster than we procreate. So the notion that we can engineer ourselves to be resistant to pathogens seems rather ludicrous from this perspective. Do you have any thoughts on this? Like, speaking, I guess, of the CRISPR babies, this idea of HIV, being able to engineer in
Right.
immunity to HIV.
Right. Well, I want to leave it to scientists to explain in detail, but very roughly, a virus invades your cell and it co-opts your machinery to make more copies of itself. That's why scientists say a virus isn't really alive. It's just like a little parasitic piece of information. It needs your cellular machinery, your ability to make proteins in your cells to copy itself. Like it needs its proteins. All it's bringing in is some information. It needs you to copy it. And so this scheme of George Church's would change the copying mechanism. Change the way your cells make proteins just by, there's like a molecule, it's called like transfer RNA. It's like one piece of the sequence. For the virus to access that, it needs to have certain genetic letters, but you could just change those genetic letters in humans so that the virus literally could not reproduce. And so in principle, it would just be, it would stop all the viruses cold, and there would be no ability for them to evolve because they would just never have a chance. They would never have a chance to reproduce even once. But of course, nature is clever. And so I agree with the speaker, if there's a way, nature is going to find it, right?
Out of curiosity, a question from Vivian Wu. Did you originally plan to, be a real lab scientist? You studied physics, or was scientific journalism always your occupational goal? How did you decide that that was the path for you? Did you have a eureka moment, a journalistic eureka moment?
Well, actually, my mother suggested it. Yeah, which is good. I mean, who knows you better? Like a lot of science journalists, I'm sort of a science dropout. I dropped out kind of early. I was in physics. I never went past my bachelor's degree in physics. So most scientists are science dropouts. Some of them go on to get their PhD and then become science journalists. I was casting around for something to do, and my mother just suggested to go to this journalism school. I would say at first I thought I would do policy, right? Those that can't do policy. I thought I would be like some kind of policy wonk, but then I just got involved with one story, and maybe I broke the first story, and then that was like getting bitten by a vampire. After that, I was like, there was no stopping me. That was the same feeling I wanted to have over and over again. So I found a calling, a little bit by luck and by happenstance.
Speaking of your calling, you reported quite a bit on the anti-aging field, where big tech companies are pouring huge amounts of money, billions and billions. Do you have a sense of what Google's Calico, for example, and others are doing? And also, just do you have any due diligence ideas? There's so much terrible reporting about the anti-aging field, articles that really border on hype. Do you have any thoughts on the kind of due diligence that journalists need to be doing? So kind of a two-pronged question.
Well, it's like the fountain of youth. Of course, everybody wants to believe it. So, is it true? Who knows? There was a story that we did a little while ago about a company called Altos Labs, which is like amazingly well funded. They started with $3 billion. Jeff Bezos is one of the funders, supposedly. And we kind of broke that story because it involved some of these genetic engineering techniques. And so that's how we got wind of it. And they actually want to use, CRISPR related technologies to actually rejuvenate people. So rejuvenation is on the agenda. And that one's interesting. I mean, it's unproven, but there is a biological basis for it, right? Like a rejuvenation is a proven technology. It's called human reproduction or animal reproduction, right? Two people older get together and they make a young person. So it is one of the most reproduced experiments of all time. So biology can do this. And scientists don't even know really, if there's a reason why people have to age. Like aging itself might just be sort of an accident. People who've reproduced are not necessary, just let them age. So there doesn't seem to be necessarily a biological reason why people have to get older. So that is quite intriguing. And genetic engineering might be a more powerful approach than, say, getting a bunch of vitamins.
Interesting.
Yeah.
There's a couple of questions about, how you get your stories. And I guess maybe this is a good time to ask you how you got that CRISPR baby story. Was that a pretty typical way that you did it, or what was there?
I mean, what is the phrase? Luck favors the prepared, or something along those lines. I mean, that's really what it is. You're spending all your time reading scientific papers, talking to scientists, and often just trying to see what's right in front of your nose, right? I mean, you can't, it's so close, it's so obvious, but you can't see it. So it's often the struggle to just see what is obvious. But a lot of times in journalism, you get a lucky break. You call someone at the right time, you hear something, and it's just sort of having those antenna up. It really comes down to often just having your antenna up and just grabbing that luck when it appears. And in the case of the CRISPR babies, concretely, it was a lucky internet search where I hit upon the documents. The guy had given me his name, he'd sort of given me his premise, and I just combined those two things in an internet search, and up came the proof.
The clinical trial documents, was that it? Or
correct, clinical trial documents. Yep, exactly.
And they were in English, or do you read?
No. Were they in English? No, they were no. Some of them were English, some were Chinese, and I had an incredibly busy afternoon. Luckily, I had gone with this documentary crew to China, and so I was able to recruit someone from that crew immediately to get the documents translated, to get a second translation from a native speaker, to figure out what they meant. And then there was also a scientific document inside this clinical trials database entry. There was a kind of a scientific document that I was able to interpret with the help of scientists, but also a little bit on my own from prior work that I had done on the technology. So I was lucky in the sense that I had all these extra resources, scientific experts I could call on, Chinese experts, and I had them because I'd been working on this, looking for this, and so it took, to answer your question, it took a team to put it together.
Genetic engineering has also been hailed as an essential tool in the fight against climate change. Is this greenwashing? Is this hype? What's your take?
I don't know. This is sort of the GMO story, like some kind of GMO crops. Look at what people do and not at what they say. Companies, Monsanto, Bayer, whoever it is, they'll often talk about, we're going to make crops that are drought tolerant or that, have bigger seeds or something. Those things are possible. I haven't really seen those yet. The killer technology is plants that survive the herbicide, Roundup that you're spraying on field. That is extremely useful to farmers. That's extremely useful. Like they can go play golf. They don't have to be out there weeding the whole time. It's extremely useful. And it's extremely, profitable. I guess the only thing in genetic engineering genetic engineering of plants that interests me is genetic engineering of the photosynthesis pathway, right? The cell, they photosynthesize, they have a whole system for turning sunlight energy from the sun into cellular energy and growing. And modifications to that process could be kind of significant. Because it's just like a solar panel. The solar panel is only 20% efficient, the plant, don't quote me on it, but I think plants are much less efficient. I think like 5% efficient or something like this. So there's a huge potential gains in how much energy plants can make from the sun if you engineer them.
Interesting. So we're nearing the end, and we've been pummeling you with questions, but I kind of want to end on with two questions. But the first is your talk's entitled "Dangerous Genes," and there obviously are other kinds of genetic engineering that aren't as dangerous, so to speak. But can you share a genetic discovery or a story that you reported on that unilaterally gave you joy or mirth? What's the most outlandish gene editing story you've actually encountered?
Joy or mirth? Oh, geez. I don't know. It's really, anytime you're playing God, it's almost always controversial. What comes to mind, we had a story about some beagles. They gene edited the beagles, but of course, then the dog people were all upset. There was a neat one about genetically engineered cows, like dairy cattle that wouldn't grow horns. Dairy cattle have horns, and so they have to be cut off. And so this cutting off of horns process is not that, it's called polling. It's not that nice to, it's done with acid or shears or something. It's kind of not nice to the cows. And what they'd figured out the genetic no horn recipe. So they could make cattle that had no, Holstein milk cows that just had no horns. They normally do. So that was a kind of a nice use of gene editing, like, improve cattle and sort of get rid of animal, veterinarian or animal problems of the dehorning. So that was a nifty one. It hasn't really moved forward successfully.
Interesting.
Yeah.
And then on a last topic, your audience here is mostly scientists and journalists, and there's something that both of these groups share, and that is that our currency is facts, right? We both really believe strongly in facts. And the other thing that we share is that the public increasingly doesn't trust either of us. So I'm just wondering if you have any thoughts about what both of these groups can do to regain public trust, and also any thoughts about whether there's any missteps that we've made to exacerbate distrust.
Yeah. This is obviously a huge problem. We have social media. Everybody gets to communicate with everybody. And it's exhausting as a journalist to try, to go through all that information. Like, I would rather be able to rely on certain sources of information as being true rather than go through all the primary information, through 1 billion tweets to try and figure out like who's for real and who's false and stuff. It's just incredible amount of work. But I have a lot of anxiety about the type of, you call it censorship or blocking or, demoting that occurs on social media in the United States. Different people have been kicked off because they telling lies about vaccines. But those procedures have at times caught up my own articles. Like, I had an article early on that was just called like how to prep for the coronavirus. And it was basically like all the stuff that you should, this is before it was declared a pandemic and before people actually rushed to the stores. I told them, go to the store now and grab all this stuff. And I guess the article was considered antisocial or stuff. I think it was like excellent advice. I took it, but it was kind of, it was corralled into a whole bunch of articles that were banned from Twitter. So once you get bitten by censorship yourself, you take quite a different view. Because it's so arbitrary. You don't know, it's just a machine that does it to you. Nobody's called to account, you can't find out why. So I weigh that against, all the false information, and also taking into consideration what's happening in China, where there's really not freedom to discuss different topics. So our system is in trouble, but is it still the best one? I don't know.
guess we should probably leave it there at the existential problems of our time. Yeah, I would just really like to say thank you for coming and giving us your thoughts and your lecture and your time. So
Yep.
I'll pass it back over to Dean DeRosa.
Great, thank you.
Wow, thank you so much for delivering our 2022 Discovery Lecture, Mr. Regalado, and weaving these stories together in such a compelling way. As a scientist, from our perspective, we tend to view, the pursuit of knowledge as something noble, a noble act. And it's so interesting to think about dangerous knowledge and unintended impacts. And really, it was so enjoyable for us. Also, thank you, Professor Everts, for all your effort in bringing this annual event to life. And to all our audience members, we appreciate you joining us virtually tonight. Before we sign off, though, I'd like to invite you to 2 upcoming virtual events hosted by our faculty, both of which were free and open to the public. The first is Data Day 8.0, a full-day conference that explores the latest developments in big data, artificial intelligence, and machine learning. This year, we'll have a panel discussion about the AI revolution, how it's transforming medicine, and many other interesting topics. We hope you'll join us on March 29th for this event. And I'd also like to mention that Life Sciences Day 5.0 is coming up in May, with registration opening in early March. This event is a full-day conference, and this year the theme will be Healthy Communities. Both events offer virtual networking opportunities and will showcase work done by graduate students at Carleton. You can visit our website, science.carleton.ca, for more information and registration details. You can also follow us on social media at Carleton Science and on most platforms, and use the hashtag Carleton Science to join the conversation. Thank you again for attending our 2022 Discovery Lecture, and have a fantastic evening. Thank you so much.